For those who don't know me, my name is Olita Johnson. I'm an assistant professor in the chemistry department at MIT. And today it is my pleasure to introduce our seminar speaker, Professor H. Jane Dyson. She's the professor of integrative structural biology and computational biology at the Scripps Institute in La Jolla, California. Her journey started by getting a PhD in inorganic chemistry, where she was doing spectroscopy of heme proteins. She did a postdoc here at MIT in the Department of Biology with Paul Schimmel, and took a brief stint as the UNESCO lecturer at the University of South Wales in Australia. She eventually became a research scientist in Richard Lerner's lab at the Scripps Institute before starting as faculty at the Scripps Institute in 1988. Since starting her independent research group, she's done, amazing work studying protein structure function, but not in the way that we typically think about them or learn about them, especially in undergraduate classes. You think about a well-folded three-dimensional structure, but what happens when you have something that seems to be much more dynamic or disordered, right? And so that can be really difficult to study. So Professor Dyson has done really beautiful work using NMR spectroscopy to study these processes, and especially how these intrinsically disordered proteins can regulate signaling pathways, particularly in transcription, which you'll hear about today. I'll briefly talk about some of the accomplishments that Professor Dyson has. So from 2017 to 2021, she served as the editor-in-chief of the Biophysical Journal. In 2019, she, along with Peter Wright, was awarded the International Society of Magnetic Resonance Award, and in 2022 she became a member of the National Academy of Sciences. So without any further ado, I will get out of the way and let you hear from our speaker, Professor H. Jane Dyson.
Well, Olita, that was quite an amazing introduction. Thank you. And it's actually a huge pleasure to me to be here again. It's sort of been 50 years since I've really entered the grounds of MIT, and boy, has it ever changed. But here I am, and I'm very, very happy to see you. And I'm looking forward to hearing your opinion on my presentation. So this is a presentation about disorder, and it's particularly applied to the way that transcription factors work. And so what I wanted to do was to begin by telling you or questioning for you, how do biological systems work? And if you think about the illustrations that you see in the textbook, they are static. There's a structure, and it's the structure of the protein. But you know, and I know, and even the X-ray crystallographers know, that these things are mobile. They're fluid, they're dynamic, and the structural features are changeable. And part of that change in structure is the way that the functional, the function is actually able to go. So we wrote this thing back in 2015 that basically says, in cellular signaling and regulation, intrinsic disorder is very, very important. After that, we really got thinking about, well, how does it work? What is happening? Why is it important? And actually, there are advantages of disorder when you're talking about signaling. And the first of them is the enhancement of specificity. If you're talking about two large proteins that come together, they have a surface area of X. If you have two very large proteins with that same surface area, you've got to have an enormous amount of protein to produce the surface area of contact. Whereas if you have a disordered protein and maybe one of them is ordered, you can wrap yourself around the ordered one. And so you've got the same surface area for much less expenditure of protein. And similarly, what you have as well are biological switches. So if you have a disordered protein, that frequently is the site of a post-translational modification, sometimes many of them. And if you can modify the disordered domain, you can make an interaction that happens in the presence of the post-translational modification or not in the absence. And so binding and interactions can be controlled by these PTMs. The other thing that's interesting about disordered proteins is that the signals can be turned off. When the complexes dissociate, what you've got is an unfolded, disordered protein. And so if they're not cooperatively folded, they're much more vulnerable to the protein destruction mechanisms. And so what you've got then is a way of really very well turning off a signal, which has to be done. Multiple unfolded partners can compete for binding sites, and that's another way of controlling the function. Now, I'm going to tell you a couple of stories today. One of them is about a protein called CREB. Now, CREB is one of these very stupidly named things that tells you that it is the cyclic AMP response element binding protein. In other words, it's a DNA binding protein that binds to the response element, the DNA bit that is responsive to cyclic AMP. It's a real stupid name, but anyway, it's better than hedgehog, probably. It's a transcription factor, and it's a very simple transcription factor, which is why I'm starting with CREB. So you get a signal at the exterior of the cell. It might be a hormone or some other molecule, and the transcription factor is activated. It activates transcription. DNA is transcribed to RNA and translated into protein, and that is the consequence of the cellular signal. So transcription factors all work more or less in this very general way. Now, the structure of CREB, as I said, is rather simple. So CREB is completely disordered. It is a 1 to 340 amino acid protein that is completely disordered as a monomer. The basic leucine zipper is the DNA binding site, and it turns itself into a leucine zipper, into a double helix at the time when DNA is bound. But if DNA is not bound, it's completely disordered. These glutamine-rich domains are disordered. The kinase-inducible domain is disordered. The whole thing is disordered. Now, in fact, the structure, CREB does structure up in some cases. The kinase-inducible domain, when it is phosphorylated, binds to the KIX domain of the CREB binding protein, another one of these really creative names. The CREB binding protein has many domains, and the KIX domain binds to the phosphorylated kinase-inducible domain. This was actually the very first structure that we ever did of a folding upon binding disordered protein that's disordered in the absence of its partner and folds upon binding to its partner here. Now, the basic leucine zipper clearly is folded when it's bound to DNA, and it makes this B-zip dimer complex with the CREB, C cyclic AMP response element. Okay, so free CREB then is going to look exactly like this. No, but what happens is the two ends of the two N-terminal ends of the CREB will continue to be disordered, and it is going to recruit what we call transcriptional co-activators. And one of those is the CREB binding protein that I showed you in the last slide. Now, it turns out that because we have this lovely disordered protein here, it gives really, really nice NMR spectra. And so I'm going to show you an NMR spectrum here of the disordered domain alone. So this is Q1, kid Q2, and I've left off the leucine zipper. And this is actually an extremely nice NMR spectrum of something which is 280 amino acids in length. Now, what I want to show you in the opposite side here is that we are looking here in the red spectrum at the full-length dimeric CREB. And so we've got 2 Q1s, 2 kids, 2 Q2s, and the B zip is folded. And what you can see is that, in fact, there's no extra peaks for the B zip. There's no extra red peaks except this one, which is the C terminus, for the B zip. So that is just simply not appearing in the protein. And the full-length dimer, the red peaks sit largely on top of the black peaks. So what this means is that we can look at the disordered monomer, and it will tell us exactly what's going on in the disordered dimer. And we're not going to look at the dimer very much anymore because it really doesn't tell us anything extra. It's just not giving us such nice information. Okay, so what we want to do is we want to look at the interactions of the CREB disordered domain. I've said the inclusion of the B-zip doesn't give us any more information and lowers the quality of the spectrum. And so we are focusing on Q1, kid Q2. And the way we're going to do this is we're going to assign the NMR spectrum of individual domains and then map the assignments to the full Q1, kid Q2. Now, I want to take a digression here for those of you who might be interested in NMR. What we use for our processing in here is iterative soft thresholding. Now, this is something that is frequently used when people are working with non-linear data acquisition. So non-uniformly sampled NMR data, NUS data, is frequently using IST, and it can also be used on our uniformly sampled data, and it gives us a much nicer looking spectrum. So IST is this processing. So what we do with IST is we take our first iteration of our one-dimensional spectrum, we cut off the bit that is the highest intensity and sequester it in another file. And then we take that spectrum where that is subtracted, and we do it again and again and again until we don't get anything at all. And what we end up with is, in fact, a very much higher resolution spectrum than we might otherwise have had. For example, in this spectrum, you might not see, for example, this minor peak. So what we did then was to take the iterative soft thresholding, and we took this spectrum here, which is the normal processing, and this is the IST, and you can see that we can actually resolve quite a lot more of the signals when we look at this particular way of doing it. So just for those who might have been interested in NMR, that was a little aside. Let's have a look at the IST NMR spectrum of Q1, kid, Q2. This is Q1 all by itself, kid, and Q2. And you can see that they are very well resolved and that they occur in different regions. The signals occur in different regions. And so when we map them onto the full-length spectrum, this is the full-length spectrum. And if we map them on, you can see that the blue ones go on the black ones, and the red ones go on the black ones, and the green ones, in most cases, go on the black ones as well. The ones I want to point out to you are two of them on this. This is this region here. This cross peak is there in the single domain, but it's not there in the full-length Q1, kid, Q2. And similarly, what we have here is another peak which is very highly shifted and very, very broadened. And so that's really the only difference. It's really strange that you might see intrinsically disordered proteins where there really was no difference between whether it's in a huge, long disordered protein or in the shorter protein. So I pointed those two out, S178 and N60. We can say that the individual domains are completely disordered. The whole thing is completely disordered. There's some small peak shifts, and some peaks are missing. And what this tells us is it's possible that there are contacts between the disordered domains. And so the way we do that, the way we explore that in NMR is to use spin labels. Now, a spin label is an organic molecule that you attach to your protein, and the organic molecule has an unpaired electron. And the unpaired electron that we use is a maleimide derivative. The way you make a site-directed spin labeling is to use a cysteine residue, either wild type if you've got a cysteine in there, or a mutant. So you take the wild type cysteine out and put a cysteine somewhere else. You use maleimide or iodoacetamide chemistry to couple the molecule. That's this molecule here. This is the coupling site. And the unpaired electron then is localized to this NO bond in the maleimide. And the way this works is if you look at the protein and say, for example, you might have this is a protein complex, for example. Here is your complex, and you've put your spin label on this particular end of the molecule. And what you find is that the resonances near the spin label will disappear. So a normal spectrum might look like this, and the spin label, when it's active, will make some of them broadened and some of them actually disappear. The closer it is, the more the unpaired electron will relax those resonances so that you end up with the disappearance of the closest NH groups in this particular case. Now, the way we control for this is, in fact, we then reduce the spin label, which takes away the unpaired electron, and that gives you back your original spectrum here. Okay, so what we've done then with our Q1, KID, Q2 was to take 3 different spin label sites. One of them was C50 in Q1, C90 in KID. That's the wild type cysteine, and the spin label in Q2 is at C172. Now, it's not particularly clear in this, and people up the back might not be able to see, but what we've got here is a superposition of a red and a black spectrum. And the black spectrum is the reduced form, and the red spectrum is the spin label form. And you can see, for those of you who have very sharp eyes, that some of those black peaks don't have a red peak on top of them. And furthermore, this spectrum is different from this one, and this one is different again. So if we blow up the glycine region here, what we can see is that if we've got a spin label at C50, we've got some of these peaks have gone, and some of these peaks have gone. It's pretty much similar if we're at C90. This one's gone here, but not here. And if we have C172, there's a lot of those glycines that are missing their red peaks, which means that the spin label has broadened them and the spin label is close to them. Now, how do we interpret this? The way we're going to do this is to take a look at the per residue intensity ratio. So this is the intensity of the paramagnetic over the diamagnetic, and the paramagnetic is the red spectrum. So if we've got a big fat red peak on top of the black peak, we'll be up here at around about 1. If the black peak is there but the red peak isn't there, we'll be down here at 0. So let's look at what happens when we put our spin label at residue 50. So you can see that there's a sort of a dip here caused by the fact that the spin label within the Q1 sequence is broadening the resonances close by in the sequence. But you can see that it's also broadening resonances over here, not quite so thoroughly, but there's definitely a big amount of broadening over here, whereas the KID domain doesn't have quite so much broadening. Now, if we look at the spin label site at 172, we can see that there's local broadening, but also broadening quite a long way away down here, and very severe broadening in Q1. So what we're seeing here is, in fact, contact between Q1 and Q2, and between Q2 and Q1 and the end of Q2. Now, what does this mean? Okay, so we've got our 50 going to 50 is going to Q2, 172 is going to Q1 and the C terminus. And so what this means is we've actually got a specific interaction between these regions, except it's not stuck. What it is a very transient interaction. These contacts are strongest in the Q regions, but we really don't know. These peptides, these proteins, remain completely disordered, so we haven't actually formed a complex. All we have is transient interactions. Now let's have a think about what the function of these Q-rich domains is. Q being glutamine, and glutamine-rich domains are present in many transcription factors, and they have multiple glutamines interspersed sometimes with other amino acids and other members of the transcriptional machinery, and they are thought to promote interaction with other Q-rich domains. And we're seeing an interaction between Q1 and Q2. And so we asked, what about other Q-rich proteins, particularly in the transcription region? So we're looking at the interaction of individual CREB domains with another Q-rich protein, the Q1 domain of TAF4. Now, first of all, what we did was we looked at CREB Q1 and Q2 interacting just the peptides by themselves with each other. So we're looking at the effect here on the NMR spectrum of N15 labeled Q2 when we add Q1. And we're seeing two effects. One of them is broadening, which means that the intensity has gone down. And the other effect is a peak shift, which means that the cross peaks moved. And both of these indicate contact between the two components of this solution. Now, if we look at the interactions of TAF4 with CREB, what we find is that we're looking here at Q1 labeled, and we're adding TAF-Q1. And what we're seeing is that there's a region right here which shows a lot of broadening and also peak shift. And so we've got a region here that is particularly susceptible to interactions with TAF-Q1. Now, Q2 with TAF-Q1, you can see that the interactions are occurring pretty much all the way over it. And so this turns out to be a specificity. Interactions are in specific regions. Q1 is between 56 and 67, and Q2 is between 172 and 185, and 222 and 235. Now, these are interesting regions, and I'll show you why. The sequences of these interaction sites, there are 1 on Q1 and 2 on Q2. These are typical of disordered proteins. They're completely mixed up. They're most unlikely to form beta strands or helices, but there are some correlations in these sequences. And the first correlation we found when we looked at the likelihood that they would form beta turns. And this is we looked in the mix server from Bax's lab, and we found that there were 3 and only 3 regions where we were likely to see beta turns. Here, here, and here. And those beta turns turn up in our sequence. There's the beta turn, 58 to 61, 177 to 180, and 227 to 230. These beta turns are occurring in the same regions of the Q-rich domain that we're noticing are interacting. The other thing that's interesting is we can actually correlate the sequence of the interaction sites. So if we line up those sequences, we've got a Q at that position. This was where the beta turn was. There's a G there and a Q there. Nothing much lines up, but that's really rather interesting, especially when we consider that this is a Q-rich domain. What this suggests is that we have motifs in these CREB Q-rich domains, and the motifs are beta hairpins. And we've got Q1 going around this beta turn, giving us a beta hairpin. This one's a beta hairpin, and this one's a beta hairpin. This one was the one that we noticed in the original spectrum that was actually broadened in Q1, KID Q2, but not in the individual domains. And what we think this means is that this doesn't occur in the individual domain. So just to remind you, that was S178 and N60. These were the ones that hadn't, that had the big effects in the difference between the individual domains and the full-length Q1, KID Q2. All right, so what's next on this project? These are Q-rich domains, and other Q-rich domains in all of these different proteins. What we found was that some of these Q-rich domains show the QXXXGQ, and some of them do not. And so our next thought in this project is to determine why we're not seeing it in some of these things and why we're seeing it in some, and to understand whether this is a general trend and whether we've in fact got different motifs in these ones that'll do the same thing as these. That's what we're going to be doing next, and I can't tell you the results because we haven't done the experiments yet. So what I'd like to do is to acknowledge the people who did the work on this, the CREB. Sergei Shnikin was a graduate student, graduated a couple of years ago. Jamie Ellis was a postdoc that left a long time ago. Rebecca, this work on CREB has been going on for about 12 years. We finally got it done. And Rebecca Burlow did some of the work. I want to acknowledge Maria, who is our staff scientist. She does a wonderful job of making the constructs. And this is a collaboration with Peter Wright. And we have funding from NCI and NIGMS. And this is a picture that I wanted to show for Olita. So this is our institute here, and this is the Torrey Pines golf course, and this is the Pacific Ocean, and the other scripts is sort of down here on the sea. So we're not on the sea, we're just, but we're close. Well, that's not the end. I have some more time, and I want to tell you a second story. And this is a story about P53. P53 is one of those things that everybody loves to hate. And I hate it most of all because there are 100,000 papers in the literature on P53. Most of them are incomprehensible, and many of them are wrong. So it's one of those things that really, really makes your hair turn grey. So from Wikipedia, which is one of my favorite sources, the P53, tumor protein P53, it's basically known as the guardian of the genome, because anything that goes wrong with P53, you end up with problems. And those problems can include cancer and inflammation and arthritis and all sorts of stuff. And this is about what's known about the structure of full-length P53. It has disordered domains. It has a tetramerization domain that makes it be a tetramer out of monomers. It has a DNA binding domain, and it has an N-terminal activation domain. So there's 4 of everything. That's interesting. So if we take a look at the domain structure of p53, we have the NTAD, the N-terminal activation domain, the proline-rich domain next door to it. The DNA binding domain is structured, though it can tend to be a little bit unstable. It has a zinc binding site, which is interesting in its own way. I won't be talking about that today. So in the disordered domain category, we have a nuclear localization signal here and the C-terminal domain. And the structural information that we have, this is, I think, this is the NMR structure actually from Alan Furst's lab. And this is the tetramerization domain in tetramer form. And if it's not tetramer form, it too is disordered. So how does it work? Well, it's clear that we've got some kind of an interaction going on between the structured and disordered domains. This particular transcription factor and many others is very difficult to study because it's a mixture of structured and disordered components. And the structured bits can be studied by X-ray, crystallography, and NMR, and what have you. And the tetramer can be structured, the tetramer of the tet domain can be structurally characterized, but you can't see the disordered domains in X-ray structures. And you can look at the disordered domains by NMR, but the problem is that the whole molecule is much too big for routine NMR. And so a mixture of structured and disordered domains is problematic. How do we study this full-length protein? And that was the problem that we set ourselves to fix about 5 years ago. Now, the NMR spectrum of the full-length p53 tetramer looks pretty good. It's got some glycines, it's got some other things, but basically what you have here is the spectrum of a disordered protein. All you're seeing is, in fact, these CTDs and NTADs. The other resonances are completely broadened because this thing is just too big to see those structured domains resonances. So how do we study this full-length tetramer? Well, one way is to use a divide and conquer technique where we just use a little bit and a little bigger bit and a bigger bit and then look at the full length. But if we want to look at the full-length protein itself, what we use is an INTIN methodology to make a segmentally labeled protein. And the way that an INTIN works is it's an auto-processing protein that will catalyze their own removal from a larger protein and fold up the protein. So we've got 2 halves of this protein. This is our protein of interest, INT, and we can see that one of these, we're going to do segmental labeling, so one of them's unlabeled and one of them's labeled. This is the XTene here, this is the INTIN here, and when we mix them together, we get the XTene beautifully segmentally labeled and ligated in the middle. It's a remarkable thing. They're bacterial protein systems. There are several different ones. They're extremely difficult to use, and what you have to do is basically keep on trying until you get it to work. I tried this many years ago and I couldn't get anything to work for my system, but our wonderful Maria got it all working for the p53. So if we take a full-length p53 tetramer labeled only in the NTAD, that's what we want to get. So here's our disordered regions, NTAD, PRD. This is the way the monomer will look. And the way we look at this is to take an isotopically labeled NTAD, and we put the whole thing together with a non-labeled rest of the protein, and we do all of the changes in the tags and the removal of the maltose binding protein, and we end up with a nice labeled NTAD, and everything else is unlabeled, and this is called NTAD P53. Now this is an interesting slide, and you might think it's not very interesting, but it is interesting. The NTAD in the full-length P53 tetramer is different from whether you have the NTAD in, well, that's the next one. But anyway, this is the same. So we've got full-length P53, and that was the spectrum I showed you before. And what I've done here is taken NTAD P53 and superimposed it on the black spectrum, which is the same as this one. And you can see that there are many, many peaks where the black is overlaid by a red. And that means that resonance belongs to the NTAD. And where the black ones are not overlaid with a red, that means it probably belongs to the CTAD, the CTD, where we haven't labeled. Okay, this is the interesting spectrum. All right, what I've done here is superimposed the black spectrum, which is just a short peptide belonging to the NTAD, and the whole NTAD P53. And this thing is tetrameric. I mean, it's a huge thing. And so what we've got is a peptide and an intene labeled NTAD P53. And the things don't superimpose. What we find is T55 moves, D48 moves, I50 moves. All of these things are moving. They're different in between an NTAD peptide and the full-length CTD. And what this means is that we have an interaction between the NTAD and somewhere in the rest of the P53. So where is this NTAD interacting? So this is the way we do this is to compare the NTAD. We look at the slightly bigger one, NTAD PRD. We look at the one that contains the DNA binding domain, and we look at the full-length protein with just the NTAD labeled. So let's start with this panel here. If you see the black spectrum, that's the spectrum of the peptide. And if we see the orange spectrum, that's the one where we've added the PRD, and it's in exactly the same place. So that means the PRD is not the interaction site. But if we look at the green spectrum, which is this one here where we've added the DNA binding domain, it moves. And so the answer, the implication, is that the NTAD is interacting with the DNA binding domain. And the same can be said of this and of this. And you can see that the 312 construct has the same position as the full-length NTAD P53. Okay, so we've got an interaction between the NTAD and the DNA binding domain. That's interesting. Nobody'd ever seen that before. And so we wanted to know what happens if we add DNA. So we took our NTAD. Sorry, my typewriter has glitched here. So we take our NTAD all by itself, and we go from the peptide to the full-length P53, and it moves. And if we then add the DNA, it goes back towards it. Not all the way, but towards in the same direction. And so this implies that the NTAD is interacting with somewhere that's close to the DNA binding site. And so the way we can determine that is to actually look at the NMR spectrum of the DNA binding domain itself. So we can actually take this folded domain and we can look at its NMR spectrum, and we use paramagnetic PRE. We use a spin label to attach it to the NTAD, and we see which parts of the DNA binding domain's spectrum are broadened. And the ones that are most broadened are the ones in the DNA binding site. In other words, what we're saying here is our biophysical measurements have shown that the NTAD interacts specifically with the DNA binding site of the DNA binding domain. So we know that this is happening, and we want to know why. What effect does the presence of the NTAD have on the affinity of P53 for DNA? And so the experiment we're going to do here is fluorescence anisotropy. So we're going to label the DNA with a fluorescent dye, cyanine 5. We're going to use two different DNAs. One of them is a high affinity for P53, and the other one is a non-specific sequence. We express our P53 constructs with and without the NTAD and see what happens to the fluorescence anisotropy. Okay, so this is actually the most interesting observation. It's not an NMR observation, so I have to say this quietly. But keep it in mind. Okay, so what we have is a black set of data where we have the full length. This is not labeled for NMR or anything, it's just the protein itself. And this is a protein where we have truncated it from the NTAD. Now look first at the fluorescence anisotropy of the P21. So we've got the P21 is in circles, and we can say that our black circles are the ones for the full length P53, and it's pretty good KD, and the red circles are for our NTAD truncated form, and its affinity is pretty good. But then when we look at our non-specific sequence, what we're seeing is that the NTAD has actually weakened. It's weakened the binding of the P53 to the non-specific DNA. Now that's significant because what it says is the NTAD binding increases the specificity of the P53 for its own DNA sequences. Now, the CTD is also a very interesting region, and so we looked at the INTENE label of the CTD region, and what we found for the CTD is that the NMR is showing here that the CTD interacts with DNA outside the cognate P53 site. And so what we've got then is a series of biophysical clues as to how the P53 is actually working. And so what we're asking here is, how does P53 work? The CTAD's interacting outside the cognate DNA sequence. Acetylation abrogates the interaction. I didn't go into this. NTAD interacts with the DNA binding domain. DNA abrogates this interaction. And cognate DNA is much more effective at removing the NTAD. So the NTAD is weakening the interaction of non-specific DNA with the DBD. So we come back to our cartoon here. How does P53 work? And you can see the cartoon says we've got NTAD very disordered and CTD very disordered, and we've got some ordered stuff in the middle. So let's look first at what happens when we have an interaction with non-specific DNA. Now the non-specific DNA can't compete off the NTAD. So the NTAD is sitting here and sequestering the DBDs from the non-specific DNA. As well, the CTD is interacting outside with the non-specific parts that are outside any binding site. And what it does is keeps it all on the DNA. The CTD is then interacting with non-specific DNA to promote the sliding of the P53 and not allowing it to pop off and get lost. Now let's see what happens when we have encounter a recognition element. This competes off the NTADs, so the NTADs are sitting away from the DBDs now. The CTDs are still holding it on there, but the main point here is that we have a DNA bound DBD. Now, the NTAD recruits transcriptional coactivators such as CBP and P300. So this particular interaction means that we are now bringing an acetylation enzyme close to the P53, and the C-terminus becomes acetylated. So it falls off the DNA, the NTAD's off the DNA, and the DNA binding domain is binding very close to the P53 recognition element. And this is the way we think the disordered domains of P53 are working to make sure that the P53 only binds specifically to its own recognition element. And so once again, I want to acknowledge the people who did this work. This is the work of a very, very talented graduate student called Alex Croyce, who graduated around about 4 years ago and promptly went off to law school, which I was extremely upset about because he was a very, very good scientist. But it turns out he's a good lawyer as well, so that's okay. Once again, I want to acknowledge Maria. She is absolutely amazing at designing sequences and designing constructs. And Peter is my husband, as most of you know, and we collaborate on many things, including 2 children and 2 grandchildren, and all of the work that I talked to you about today was a collaboration with Peter. Okay, so we have a couple more minutes. Would you rather ask questions or would you rather learn something else? What would you like? Nobody's saying anything. What? Questions? Learn. Okay, here we go. I'll do it quickly, and you can ask questions because it's probably going to be too quick. I'm going to tell you a little story about the phosphorylation of p53. Now you'll remember this slide. We've got our folded domains and our unfolded domains here. Now it turns out that phosphorylation is a huge controlling feature of the way that p53 works. And one of the phosphorylations that happens is at T55. Now T55 is actually a constitutively phosphorylated phosphorylation-dependent regulatory switch. It's constitutively phosphorylated, and in DNA damage, which is when p53 is turned on, it becomes dephosphorylated and leads to transcription. And then when the reaction has to be turned off, it's rephosphorylated and everything goes back to normal. And the way we had to do this was actually very interesting because we have to, we use casein kinase 2, but casein kinase 2 phosphorylates 4 different phosphorylation sites. And so we have to assign the resonances and so forth. We can get an NMR spectrum. I apologize for the quality of this. It came out of somebody's thesis. This particular area is where all the phosphorylated serines come and threonines. Otherwise, they'd be up here. Okay, so I'm not going to go into this in great detail. We were able to use by mutation, we were able to get the assignments of all of these things. So we're going to do this by looking at the mutation of the phosphorylation sites. So we're going to make 3 mutants. We're going to make an A3 mutant where we've taken 3 of the phosphorylation sites, S6, S46, and S392, and turned them into alanines. And that one, where we have that one, will only phosphorylate, your CK2 will only phosphorylate T55. In A2, we have S46 and T55, and A4, the control has none. And you can see that, in fact, we can make sure that we've got two phosphorylations in our A2, 1 phosphorylation in our A3, and nothing at all in our A4. So if we add DNA, what happens? Well, for a start, if we have PT55 all by itself, then it just sits here. But then if we add DNA, it goes up here. So addition of DNA has changed the NMR spectrum. And if we have A2, that will also happen as well. Okay, so then the most important thing that I wanted to tell you was to ask how phosphorylation is affecting DNA binding. And so we use fluorescence anisotropy. We use Cy5 labeled cyanine 5 labeled DNA, and we use 2 sequences. Now this is a bit different from before. Here we have the P21, which is the high affinity, and this DINP1 is actually also a P53 site, but it actually is a lower affinity site. It does the activation of the P53 regulated apoptosis genes. And so cell cycle regulation, P21, has a higher affinity. And then we add P53, 2 mutants, where we have our A3, so that's only got T55, and then we have our A2, where we've got the 2 phosphorylation sites. These are P53, and these are unphosphorylated. These are phosphorylated. So remember the colors, blue and green, unphosphorylated, black and red, phosphorylated. Right, let's look at the blue and green ones first. If we look at the blue and green for P21 and for DINP1, the blue and the green, that's unphosphorylated A3 and unphosphorylated A2, they're pretty much the same. Higher affinity for P21, lower affinity for DINP1. Now if we look at phosphorylated A3 and A2, look at this. I didn't believe this when I saw it. As you add more and more and more and more P53, your fluorescence anisotropy indicating binding goes up and it goes down again. And this one goes up and down to 0, and this one even more so. A2 is more affected than A3, and A2 is the one with 2 phosphorylation sites. So how are we talking about here a biphasic behavior? Well, one of the ways we can think about this is to think about what the binding of P53 is. And the P53 is binding, the phosphorylation of T55 actually enhances the binding of the disordered NTAD to the DBD, and it competes with the DNA. It's even competing with the DNA for high affinity DNA sites. But what's the source of the biphasic behavior? Well, the source of the biphasic behavior is this. The phosphorylated P53 is binding to DNA as a dimer. The first dimer binds, and so the curve goes up. But it's phosphorylated. And so when you add a second dimer, there's too much negative charge to bind to the DNA. And so the unfavorable negative charge kicks everything off. And so you're kicking off the first dimer and the curve's going down. Now, so what this means in physiological terms is that the constitutive phosphorylation of T55 is preventing the formation of the complex of tetrameric P53, that's the active complex with the DNA, with its cognate DNA. And the reason why you might want to do that is because you don't want P53 acting when it hasn't had a signal. It has to be inactive unless it gets a signal. And when it gets a signal, the T55 is dephosphorylated and it can work properly. So this is actually something of a theme, that the post-translational modifications are frequently changing the charges. And you can see the logic of this because P53 is binding to DNA, and DNA is a very, very highly negatively charged molecule. And so when you add negative charges, you definitely lower the affinity. And so the people who did this work were, again, Alex Shunsun, who's amazingly creative postdoc, Sang-Ho Park, who did the CTD work, Maria again, and Peter, and once again, the beautiful scripts. And thank you for your attention.
All right. Oh, there we go. Thank you for the amazing talk. I realized that I forgot to thank the people that sponsored this seminar series in the introduction, so thank you to those people for sponsoring those entities. And with that, I'd like to open up the floor for questions with trainees asking the first question. So if you're a student or a postdoc and you have a question, raise your hand and we'll bring you a mic.
Up the back. Oh, he's coming down. I put up your sponsor slide, so.
Ms. Burke, thank you. Very nice talk. I was wondering what, after looking at your slides and your presentation, of the P55 and the P53 interaction. So what would be the eventually application for like therapeutics, like application in the context of inflammation and cancer? Because we all know the interaction of P53 with DNA mutation. So what's your opinion and future perspective of this?
You know, that's a really hard question. From my point of view, what I'd like to know is how it works, and then I'm going to think about how I can disrupt it or fix it or something like that. But we've really only just done this, and so I haven't really thought about therapeutics at all. It's a very good question, but you know, you guys are really much more into that kind of thing than me. I don't know, you know, constitutive phosphorylation, that's going to be very hard to do a therapeutic on, isn't it? But it's nice to know how it works.
Yeah, definitely. Thanks.
I mean, if it is getting phosphorylated and getting constitutively activated, or dephosphorylation actually helps in activation, right?
Dephosphorylation causes the activation, yeah.
Right. So then one can think of making bifunctional molecules to dephosphorylate P53, and that can cause, in a very maybe cancer-specific activation.
Well, I think what you have to worry about there is that P53 acts in so many different situations that, you know, you can muck it up for the cancer, but unless you can kind of inject it right into the cancer, you're going to muck up everything else as well. This is the real problem for cancer therapeutics. And I mean, you know, the poor people with cancer, they take chemotherapy and it wrecks their physiology for a while. And I just feel like, you know, we can, the understanding will help us, but I don't think we're there with a magic bullet yet.
Hi, I'm here. I really like the creative use of Intian ligation. I thought that was like really nice to label the one domain to like isolate those interactions. I guess I was just wondering, because I know some Intian ligations leave scars, and so I was just wondering what was the choice of Intian ligation versus like native chemical ligation, or
native chemical ligation is very difficult. Particularly, part of the problem is that the bits are not stable on their own, and that's why, I mean, that's one of the reasons why it's so difficult to get inteins working, because sometimes the intine, extine protein that you make just crashes or doesn't express or something like that. That's why I said it took a long time for us to get, I mean, I don't know whether you noticed, but there were tags here and tags there, and some of them were solubility tags, and some of them were expression tags. I mean, it took a lot to get that intine thing working. Whereas when I was doing it, I was like, okay, we'll put this one on here and this one on here. Well, nothing expressed, you know. It's very cultural. You've really got to get in there and work on it. I would say it's probably one of the most difficult things we've ever done. But don't let that discourage you, because, you know, to be perfectly honest, sometimes, you know, we're sweating away and somebody comes up and goes, oh, but that worked for me right away, you know. I mean, it's like, it just depends on your system, yeah. Yes, we did. I think there are now sort of 5 or 6, and if one doesn't work when you've tried everything you could think of, another one might.
We have another question in the back, and I'll just note that I am monitoring the chat so for people that are live streaming, if you have questions, go ahead and put them in the chat.
Hi, thank you. That was a really illuminating talk. I'm just wondering, going back to the CREB components, you mentioned from the spin labeling that you knew that the interactions were largely intramolecular, and I was just wondering how you came to that conclusion.
How I knew that.
Yeah.
The information was actually on one slide, but I didn't go into it. The way we did it was we had one labeled, we made 2 samples of the Q1 kid Q2. One of them was isotopically labeled and the other one was not labeled at all, but it had the spin label. And so then we mixed those two together, and when we saw a spin label effect, it would be intermolecular in that particular case. And this, I actually showed the data, but I didn't mention it because I didn't think anybody would be so sharp as to want to know why. But yeah, that's how we did it, yeah.
Thank you so much for the talk. I'm just curious about the Q-rich slides you provided. Does it matter how many amino acids in between the Q and GQ, and how do you think this will impact the protein-protein interaction?
I don't know the answer to that. It's an extremely good question. At this point, we just published this a few months ago, and then we started to think, well, you know, what next? And that's when I did that sequence alignment. But I mean, the fact is, all of those other Q-rich regions do interact. So maybe the different, you know, maybe the ones that I showed all had 3 in between, but it may be that there are some with 4 that I missed, and so, you know, there's a lot of work to be done in the sort of bioinformatics there, which we haven't done yet because we're still floundering around. But it's a very good question, thanks.
We have a question in the chat from Ed Skolnick, who asks, have you introduced mutations in P53 that occur in cancer and then studied the structural biology that you're doing?
No. In a word, no. What we were interested to do was to try to understand how P53 works normally. I guess perhaps one of the reasons why we haven't done all the cancer stuff is the 100,000 citations and reprints and stuff. We probably will look into that. We haven't been interested in it lately, simply because so many other people are interested in the cancer. And it just seemed to me that finding out how it worked may not necessarily give us a therapeutic, but it may let us understand a little bit better how it works.
I'll exercise moderator's privilege,
Yes.
And ask a question. So actually, going back to your CREB story, I noticed that there looked to be a few serines and threonines in those beta hairpin motifs you pointed out. So I was wondering if those are post-translationally modified and if you see them affect the stability of those beta hairpins ever, or if you've
That's
looked
a
at
very sharp eye, and no, we haven't done any of those works at all. It is quite likely that some of them would be, if they have the right sequence surrounding them. But we really haven't looked into that yet. It's all sort of new stuff.
Thank you. Any other questions from the room? All my students in 554, we just talked about transcription, not one question.
Oh, there's one back there, yay.
Thank you. Great talk. Just quick question. If you're working with transcription factors and whatnot, do you find it difficult at all to express any of them? Like, do they sequester any of the DNA in the organism that you're expressing them in?
No, they don't actually. Because we're overexpressing them, you know, it's just, I mean, basically we're using the E. coli, which we use as a factory. And so we grow it up to OD1, and then we just throw in the label and induce and basically those poor little guys just die, you know, but not before they've made us a whole lot of protein. So,
thank you.
Sad.
Any other questions? I'm coming.
Do you have any comments?
Wait, we can't hear you if you don't speak in the mic.
Thank you for a wonderful talk. So do you have any comment on the phosphorylation of serine 46 in P53? It seems that you have a case that both serine 46 and threonine 58 were phosphorylated.
55, yes.
55, sorry.
Well, they were phosphorylated in our system. We wanted to understand, serine 46 is also phosphorylated, but not constitutively. So, you know, I mean, it's very complicated, the phosphorylation business, and off the top of my head, I can't tell you when it's phosphorylated, but it certainly is one of the important ones. I think Shun did that because he wanted to understand what happened when you accentuated the charge. I think that's the main reason he did that. But there is no doubt that the serine 46 is phosphorylated during the cell cycle. Yeah.
Yeah, it seemed to be very complicated study.
Yes,
because
it's
it
complicated to me too.
In some study, they report that phosphorylation serine 46 like activate P53, and here we see some in different ways. So I
inhibition,
think
yeah, it's weird, isn't it? Yeah.
It's really interesting. Thank you.
Yeah.
Any other questions?
Thank you for the talk. I was just wondering if you were going to do further assays to try and characterize how exactly, this is for the CREB story, how exactly like CREB is interacting with the DNA, like now that you know that there are peak shifts when you added the DNA through like the NMR data, like are you gonna look further into that? Like do you have plans?
CREB. No, we didn't do a lot with the DNA in CREB. I think you're thinking of the P53?
Oh, maybe I am.
Well, never mind. In CREB, you have the B-zip, the basic leucine zipper that comes together and binds to DNA. And that's pretty well known. I mean, we didn't really look at DNA binding in CREB because what's happening. But, you know, yeah, whatever. Sorry, I'm getting tired.
All right, and this will be our last question.
Hi, thank you for the talk. I'm curious, like you showed the NTED helps to recognize the correct target DNA sequence.
Yes.
I'm curious, how does this mechanistically happen? Like,
Okay, yeah.
it's obstructing the DNA interface in case of non-specific, but
Yes.
it just moves away when there is a specific sequence.
Yeah,
wants to-
it's really more a competition. So you can think of the binding element, the cognate site, as being stronger than the NTAD. So it can replace the NTAD because it binds more strongly, whereas the other one can't because the NTAD wins the battle, if you see what I'm saying. And so when you get the cognate element, we don't know how this works. We just see what happens in the test tube. But those NTADs go off, and they can't go very far because they're still attached to the DBD. And so what we then think is that they go off and recruit the CREB binding protein and the P300 to do the transcriptional activation.
All right, with that, I'd like to thank Professor Dyson again for the amazing talk. Thank you for being an engaged audience. And happy Monday.